Hello, everyone. Today we're going to look at the Lac Operon, which is, as hopefully you'll have come across already, how bacteria um, sort of group together their genes into sort of one functional unit, which we will see in a minute. But if you need a recap on that, go back to the gene architecture lesson, which I think was lesson 1 for the upper sixth. And you can recap the difference between prokaryotic and eukaryotic gene architecture. But prokaryotes use what we call operons. So I'm going to jump straight into point number 1 here, which is to define what an operon is. And again, hopefully you have covered this already, but I'm just going to make it clear. So definition. So this is number 1, definition of an operon. And the definition of an operon that you need to know is it is a group of genes, that are placed under the control of a single promoter. And the sort of next bit, which is sort of equally as important, although it's not required explicitly for A level, is just to make the point that these genes have related function. So I'm going to sort of add just a sort of asterisk here for a little bit of information or a little bit of extra information, which basically says that these genes that I have referred to in my definition up here, these genes have related function. So the genes in question act usually in a similar or the same biochemical pathway. So let's just take a sort of extended look at this, sort of from a bird's eye view. If we have a substrate and we turn it into this product here, on the right-hand side, actually I'll draw that a bit bigger. It might be that it is not what you would colloquially sort of think of as one step, catalyzed by one enzyme. It is more likely to involve multiple steps with multiple intermediates. Here, to eventually get to your product, which is over here in yellow. And each of these steps, you could imagine, require one, maybe more enzymes. So you'll have this sort of blue enzyme here working to sort of act initially on the substrate. You then might have another enzyme over here, another one here, and another one here. And all of these enzymes, enzyme 1, 2, 3, and 4, are required to convert our substrate over here into a product over here. It is multi-step, which is generally how biochemical pathways proceed. So, in terms of being a bacterium, you could imagine that because their genomes are quite small, comparative to certainly higher eukaryotic organisms, they need to be far more efficient with how they structure their genes. So they use this idea of an operon to ensure that all of these enzymes, the genes that code for all of these enzymes, so this one, this one, this one, and this one, are placed under the control of the same promoter. And the promoter, you hopefully can appreciate or remember, is the site in DNA just upstream of your coding sequence that acts as the binding site for RNA polymerase, which will be the enzyme that actually carries out transcription. So essentially, all of these enzymes will be expressed together. We will make all of these enzymes essentially at the same time, simultaneously. And that is because they will be transcribed into a single piece of mRNA, which we will see. So that's the first thing. We have defined and sort of briefly recapped what an operon is, so I will tick that off the list. And now we're going to look at the general structure of a sort of generic operon. So point number 2. I'm going to now draw my bit of DNA here. Hopefully that converts into a straight line. Yes, it does. So I'm going to draw my sort of, don't forget we're in prokaryotes here. We're sort of talking about bacteria. So I'm now going to draw a prokaryotic operon, just a sort of generic one. So in green at the start, we have our sequence here, and that is the promoter, which is where transcription will be initiated from, typically from an adenine residue at the end of the promoter there. But this is where RNA polymerase will bind. So in orange here, if I just convert to orange, this is RNA polymerase. And that is going to move along the DNA and transcribe our genes. Of course, it's important to always label our primes as well. So RNA polymerase will move in a 5' to 3' direction. That's how it will synthesize the RNA. So downstream of the promoter, we then have our genes. So let's do one in purple here. I will then do one in blue. And let's go yellow. And of course, at the end of all of this, we have our terminator region. So I'll do that in red. I'll just do it as a dot here. So this is the terminator, which is kind of the sort of functional opposite of the promoter. It is the signal that tells RNA polymerase to dissociate from the DNA, and release the mRNA product. So let's look at these 3 genes. Of course, at the start of all of these, we will have our coding or start codon, which, if we're talking about the DNA sequence here, would be ATG, read from a 5' to a 3' direction. But each gene has its own start codon. So, of course, we need to put the one in blue here. So this would be ATG, and the same in yellow. And at the end of each one, we have a stop codon, one of the 3. So I'll just draw that as an S for simplicity here. So S refers to one of the 3 stop codons that we have available to us. And the important thing is that at the beginning of every coding sequence, so each of these genes represents a coding sequence, right, where you can sort of start your coding information from, and where you end, that whole intervening sequence is called the coding sequence. So this is an uninterrupted bit of coding information to make our protein. So it can be read by the ribosome and sort of translated as a unit to make a functional protein. So it is therefore called a coding sequence, or CDS. Same for the blue, same for the yellow. But I'm going to get rid of that just for the moment. The important thing is, how does the ribosome know to do this? And the answer to this question, if I just work out, there we go, we're fine, is that we have a ribosome binding site just upstream, just in front of each coding sequence. I'm doing it in purple here. So if I can switch to a sort of more appropriate purple color, here we go. That is a ribosome binding site. As is this, and as is this. So our ribosomes know where to bind once we've transcribed into messenger RNA. And that's essentially our operon. We have gene A, if I swap color again, gene B, and gene C. And this is in DNA, don't forget. Because we are talking about genes. And a gene, as we all know, is a section or a sequence of DNA that encodes for a protein or a functional RNA molecule. So, if we now carry out the process of transcription here, we are going to produce our messenger RNA. And of course, in bacteria, they don't have to edit it. They don't have to put a 5' cap on the 5' end. They don't have to add a poly A tail, and they most certainly don't have to deal with any introns. So we're going to start transcription where the promoter is there for the initial residue, the A there that's going to be added. So we start here. So we're going to make our mRNA. I'm just going, actually, I'll do it as I go. We'll then have the ribosome binding site, number 1, ahead of our first gene, which in this case is gene A. We then have our next ribosome binding site almost immediately afterwards, and we then go into gene B, and we have our next ribosome binding site as we enter gene C. And of course, we have a little bit of untranslated region either side. So hopefully you can appreciate that is what our mRNA, mature messenger RNA, is going to look like. And hopefully you remember from last time, the bits sort of upstream of the genes and downstream, this is a 5' region in here, and the 3' region here, are known as untranslated regions. Or UTRs, if you come across that term. Don't worry too much about them. Key thing is you've got ribosome binding sites ahead of every single gene, and each of these genes has a start codon and a stop codon. So what can happen now is as you are transcribing, your ribosomes can bind here, typical 2 subunit ribosomes here, to your ribosome binding sites, and they can then start to translocate along the mRNA and start making our proteins, either A, B, or C, and they are then translated, and we get from this our 3 proteins. One might look like that. One might look like that. Horrible drawings, but I hope you get the point anyway. And the C might look like that. So hopefully that has now covered the second point, which is the general structure of an operon, how it works. And don't forget, transcription and translation in bacteria are coupled. As soon as you are producing that mRNA, you can bind ribosomes to it. The final thing, just before we move on, is just to give one more word to you that you don't need to worry too much about, but I think it's probably useful for you to know it anyway, which is an mRNA molecule such as this that contains coding sequence for multiple genes and multiple products, in this case, is known as polycistronic. Polycistronic. And a cistron, so just to sort of explain this word, it probably makes more sense to explain it than just sort of leave you hanging. Poly obviously means many, and a cistron refers to a gene. It's just a sort of an alternative term for a gene. So polycistronic means essentially multiple genes on the same mRNA molecule. So let's take that off. So general structure of an operon done. We now need to look at a case study. And the case study that we are going to look at is that of the Lac Operon. So this is point number 3, which is our case study for, certainly for OCR A, the OCR A syllabus for A-level biology, but possibly for others as well, which is the Lac Operon. This was best studied in E. coli. And we know a lot about how bacteria regulate their genes from this particular system. And the aim, or the sort of overall point of this system, is to produce the gene products that are required, of which there are a number of them, for metabolism of lactose, for metabolic processing of lactose. Which brings us all the way back to our carbohydrates from lower sixth, biological molecules, because we're just going to do a quick recap here. Hopefully you remember that lactose is a disaccharide, and it is made from the two monosaccharides. If I just draw sort of generic hexose sugars here, hexose meaning 6 carbons, don't forget. I'll draw another one just to make it clear that they are slightly different, and they are joined by a bond that looks like that. That bond here is called a glycosidic bond, and it is formed from the condensation reaction between two monosaccharides. So we liberate water here. If we were to produce this, we'd also produce water, or a molecule of water. And this lactose here, this disaccharide, is made up of two monosaccharides. In purple, we have glucose, and in black here, we have the isomer of glucose, the stereoisomer of glucose, which is galactose. Again, recap your lower sixth biochemistry if you need any more information on that. I'm not drawing the full structures because you don't really need to worry too much about it. The main structure you need to be able to draw, of course, is alpha and beta glucose, those two isomers, but you are not required to be able to draw galactose in any sort of detail. So I'm just going to stick to sort of drawing them as generic hexagons here, but also just to point out that they are, of course, both hexose sugars. They both have the formula of C6H12O6. Okay, these are isomers of each other. And again, I realize this makes these videos sort of a bit longer because I go on these sort of detours, but I hope it's useful to you when you're revising, because you can then revise some lower sixth content whilst doing upper sixth stuff. So the key point is, how do we metabolize this? And the answer is we need to cleave this glycosidic bond here down the middle. Oops. Oh, that's rubbing it out. Okay, fine. We're sort of cleaving it in a weird way. So if we essentially cut that bond, we then liberate glucose and galactose as individual monosaccharides. And if I redraw them down here as separate monos, like this, hopefully you can appreciate that the glucose in purple here can immediately be metabolized by respiration. Again, just pointing to sort of upper sixth topics, of course, this immediately will be a substrate for glycolysis, which is the first step of respiration. More detail on that in the respiration video that will be up at some point. The problem with galactose is it is not directly able to be metabolized in the same way that glucose is. We cannot do the same thing immediately. Because it is a stereoisomer, it is not a terribly good substrate for the enzymes that act in glycolysis. The first one, of course, being hexokinase. And essentially, we need to convert this into a sort of intermediate product that is able to be metabolized before we then can sort of put it into glycolysis. But we're not going to worry too much about that, but just be aware that that does happen. The key point is we can liberate glucose, put that into respiration, produce our ATP, off we go. So the next point that I wanted to make regarding that is that glucose doesn't have to be put into glycolysis. So this is just something to bear in mind. This glucose molecule over here doesn't have to be put into respiration. It could be put into a number of different pathways. The first, well, you don't need to know them. So actually, I won't tell you. Otherwise, we'll be here all day. But what I will say is, of course, that you can immediately start putting this into glycogen. I'm only going to draw a few here, but hopefully you get the point. You can start packing this together, horrible hexagon there, into glycogen. That requires separate enzymes via separate pathways. But the point is, we liberate glucose from lactose by cleaving that glycosidic bond using hydrolysis. So essentially water is going to sort of re-emerge here. So water is going to act as a nucleophile here, one of the lone pairs, and it's going to attack that bond and it's going to cleave it and break it. And the enzyme that helps achieve this, hopefully you remember from GCSE, is called lactase. More on that in just a minute. So let's look at the structure of the Lac Operon. The whole point is we need to achieve this if there's lactose in the environment. So the Lac Operon, I'm going to draw a very similar structure to the one that you saw before. I'm going to draw, I'll do it in a slightly different way, which you'll see why in a minute. So I'm going to draw the promoter. Actually, I might draw this underneath. Promoter here. Just downstream, we then have our genes, and we have 3 of them that we need to worry about. Now, just for the sake of ease, actually, no, scrap that. I will put the ribosome binding sites in. Why not? I'll do them in blue like that. I was going to not do that, but I will, just to keep things consistent. So another ribosome binding site here. And let's just go for a red one, why not? Another ribosome binding site here. And we have a terminator, which I know I've done in red up until now. I'll do it in purple, this sort of purple color. We have our terminator sequence. So promoter is where RNA polymerase is recruited and can begin transcription. The terminator is where essentially it is told, in inverted commas, to dissociate from the DNA duplex. So let's have a look at the 3 genes first of all, and then we'll build up the picture a little bit more. What are these genes called? The purple one here, the first one, is called LAC-Z. This is the gene name, by the way. This is very important. This is the name of the gene, not of the protein that is produced from it, or that it codes for. The yellow gene is called LAC-Y, and the red gene is called LAC, horrible L, apologies, LAC-A. And the proteins that they code for are as follows. That's horrible sort of looking enzyme. It's even worse. Oh well, we'll go with it. This is an enzyme and it is lactase. That's going to confuse things in a different color. I'll stick to purple. It's lactase essentially. So this is the enzyme that up here is going to cleave here via hydrolysis our glycosidic bond between our glucose and our galactose, liberating them as monosaccharides. However, we don't call it lactose now. We're at A level. We call it its actual name, which is beta galactosidase, which is the sort of proper name for this enzyme. I will just put lactose here just so you don't forget what it does. Lactase cleaves lactose, but beta galactosidase is absolutely the term that you need to use for LAC-Z. And I remember it because the S sounds like a Z when you say it. So galactosidase is how I remember it in my head. We then have LAC-Y, which codes for a sort of carrier protein called a permease. So I'll draw it like that. And it is called the lactose permease, which transports the lactose into the cell. from outside, and we'll get to that more in a minute. Finally, we've got Lac A. You don't need to worry too much about this. So I'm going to put a sort of cross here. I will tell you that it codes for something called transacetylase. But we don't really talk about it at A level. You don't need to worry too much about it. Essentially, it's involved in this pathway up here. I like how I tell you I'm not going to tell you these things and then do. I can't even find my drawing now, so this is going well. Did I delete it? Oh, no, it's here. Oh, maybe I did. Oh, no, I didn't. It's here. It's involved in metabolizing the galactose that we cleave off here and turning that into a useful product for us. But don't worry too much about it. In terms of lactose, you need to really focus in on Lac Z, which codes for beta-galactosidase, and Lac Y, which codes for lactose permease, or you can just call it permease for short. Beta-galactosidase, let's put a note in here. This will cleave our lactose by hydrolysis. So we need water to act as a nucleophile. And the permease allows transport of lactose into the cell. So to put this into sort of diagrammatic terms, what we're saying is if you have, or we have here, our bacterium, our E. coli in this case, so it's got flagella and it's got its sort of nucleus. Ooh dear, what is happening here? Go back. I think we're okay now. Oh no, it's all going wrong. So it's sort of DNA here, circular genome. The key thing that we need to do is if there is lactose in the environment, so I'll draw that as sort of squares here. If there is lots of lactose in the environment, we want to be able to use that as an energy source for respiration and various other things. So essentially what needs to happen are two processes. We have to transport the lactose into the cell. So we have to start taking it into the cell. That is achieved via the gene product or the sort of protein product of the Lac Y gene, which is the permease. So that's why we need the permease to be made, because that's how we get lactose into the cell. And the next thing we need to do is we need to be able to take our substrate here and convert it into our product, which is glucose and galactose. And that is achieved via an enzyme, and that enzyme is beta-galactosidase. Which is coded for by the Lac Z gene. The transacetylase we don't worry too much about at all. So that hopefully gives you a sort of diagrammatic representation of sort of why we need these two gene products. We need beta-galactosidase to cleave the lactose when it's in the cell, and we need the permease to get it into the cell in the first place. Now, just before we move on to how this is regulated, I'm just going to draw a couple more things in. I'm going to draw another binding site here, and I'm drawing it this way for a reason. I'm just picking the color. Let's go orange. There's a binding site just sort of downstream of the promoter before the Lac Z gene, and it looks like this. And it is called, not to confuse you anymore with words beginning with O, it is called the operator sequence in orange there. And hopefully what you can appreciate is that we have an overlapping bit here where the operator somewhat overlaps with the promoter. So I'm just going to write here, nota bene, there is some overlap with the promoter sequence. and that's quite important. So the whole orange box there is the operator, and it overlaps, it shares sequence with the promoter by actually a sort of reasonable amount as well. The final thing I just want to point out here is there is one more gene that is not part of the Lac Operon. I'm just going to check. I'm not going to cover that later. I can't remember now. I'll talk about it now because we're going into how it works. So the important thing here is we have one more gene, except this gene is not within the operon. It is outside of, it is just upstream of the promoter. So I'm going to draw it here in blue. Importantly, it has its own promoter. So I'm going to draw the promoter as just a blob here. So this is the promoter for this gene, and it will have a terminator just downstream in red here. So that's the terminator. And it has its own coding sequence. So it's a little bit more like a eukaryotic setup. It's not in an operon. It is just its own entity in blue here. So we're going to get transcription of this guy. And this guy is going to be transcribed and then translated, as per usual, into a protein, which might look like this. Just my generic protein thing. This gene is called LAC-I. And it codes for, so this is the LAC-I gene product, and it codes for this blue blob here, which is the Lac repressor. And we've talked about repressors when we looked at genetic circuits, where repressors cause repression of a circuit or a sort of biosynthetic pathway, as opposed to induction, which is caused by activators. So I bring this to your attention because there's one more sort of distinguishing point I want to make between LAC-Z, Y, and A, and our new friend up here on the left-hand side, just upstream of the entire operon, which is the LAC-I gene. And I do just want to use the fact that this is a video and I can sort of spend longer on it than I would in an actual lesson with you guys, just to make the point that all of this is an operon. We have multiple genes here, 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 under the control of the same promoter. This guy, LAC-I, is its own gene, its own coding sequence with its own promoter and its own terminator. It will have a start codon, don't forget here, ATG. It will have a stop codon at the end. It sort of exists on its own. And the other difference here is that it codes for a repressor. And a repressor here is a protein that hopefully you remember from your the first lesson on circuits. This will act to cause repression of a genetic circuit. Essentially, it is what we call a regulatory protein. And we therefore call the gene for this a regulatory gene. And that is the important point. It is a regulatory gene, the Lac repressor, because it codes for a protein. It's not an enzyme or anything that's going to sort of do anything like that. Its job is to control gene expression. It is a regulatory gene product. So it's therefore a regulatory gene. So inducers, if there were genes for inducers and things like that, then they would also be classified as regulatory genes. And when we look at eukaryotic transcription factors absolutely come under this bracket. So Hox genes, when we refer to this, I'll put a quick note here. So Hox genes, which comes up next, involved in development of a body plan. These are regulatory genes as well. I make that point because that is different functionally from these genes, our Z, Y, and A. These genes code for proteins, as we've seen, that do jobs that do not regulate gene expression. They carry out other functions, right? They're enzymes that cleave substrates or that kind of thing. They're not binding or interacting with DNA at all. So these are called structural genes. Structural genes. And that is one distinction that you really do need to know and make very clear. The difference between a regulatory gene that codes for a protein that interacts with DNA and regulates gene expression versus a structural gene, which is pretty much most genes you've come across up until now. So things like beta-galactosidase, insulin, various other enzymes, polymerase, helicases, antibody genes, that kind of thing. They are all structural genes. Cool. So let's tick that off. So that's our Lac Operon, pretty much done in terms of the sort of architecture. I now want to talk about the induction of the Lac Operon and how that works. So how is it actually turned on? And this is going to be point number 4. Induction. So remember, the definition of induction is the process of using sort of either activators or inducers to increase the synthesis of a gene product. So let's quickly draw the Lac Operon again. You'll probably get sick of this in a minute, but here we go. I'm going to draw the promoter a little, draw it a little bit bigger. Let's do it like that. So this is the promoter. We then have our operator sequence. I'll try and keep the colors as I did last time. So our operator, which I'll get to, which overlaps with the promoter. The importance of that we'll see now. And then we have our genes. So in this case, I'm just going to do random colors here. We've got our Lac Z. We have our Lac Y. And we have our Lac A. And of course, we have our terminator at the end. Let's just extend that a little bit. So don't forget, we've got the ribosome binding sites. I sometimes will forget to draw them in, so apologies for that. But we've got a ribosome binding site within the intervening bits between the genes. There's not much DNA between them. They're fairly continuous, but there is a ribosome binding site there. So what happens is as follows. We have our Lac I gene, which is just upstream. And I'm not going to draw the promoter and the terminator. I'll just draw the Lac I gene here. And the Lac I gene will be transcribed. It isn't regulated, actually. It's just something to bear in mind. It's not really regulated to any great degree. So we're going to regulate the Lac Operon here. So be able to control whether it's on or off. But the Lac repressor is always being transcribed from the Lac I gene. And that will make the mRNA for the Lac repressor, which will then eventually make the protein. And I'm going to draw it like this. This is the Lac repressor. And it is, as you can see here, a tetramer. Okay, we've got 4 individual subunits. So it is therefore yet another good example of quaternary structure of proteins. Okay, there are 4 tertiary structural elements. 1, 2, 3, 4. And the Lac repressor binds to, and this is the important thing, the operator, like this. Okay, so the Lac repressor binds to the operator. And hopefully what you can appreciate is, because the operator overlaps with the promoter, the Lac repressor is sort of sterically hindering the RNA or any RNA polymerase from coming along and binding to the promoter. Because what would really want to happen here is any RNA polymerase that you have wants to come along and bind to the promoter like that. So this would be your RNA polymerase. Whoops. But you can't do that because if your repressor in red there is bound, because the operator sequence that it binds to, the operator being just the binding site of the repressor, because that shares sequence with the promoter, that means if it is bound, the RNA polymerase cannot be recruited to the promoter. It cannot bind, right? It will sort of stay here because it cannot bind. The repressor is sterically hindering or getting in the way of the RNA polymerase binding to the promoter, and therefore causing transcription to happen. So hopefully what you can appreciate here is this is a system that in normal circumstances, it is turned off. That is the default position. It is off. We are not getting transcription of LAC-Y, LAC-Z, and LAC-A. We are therefore not producing the enzymes required to metabolize lactose. But how do you turn this on? And this is the clever bit. The absolute magic ingredient here, so I'm just going to write magic actually, why not? The magic ingredient is that the Lac repressor kind of actually looks a bit like this. I'm going to try and draw this to the best of my ability. It looks like this. And you can imagine that it's binding to the operator here. So just excuse my sort of fairly crass diagram here. This is the DNA in black. And let's say this sequence here is the operator, which I've shown up here slightly better. What I have drawn, though, is other sites in the polymerase that look interesting. And so how we get transcription to happen is we want RNA polymerase in green here, if I sort of draw it like this, to bind to the promoter. But it can't do it yet because the repressor is bound. So we need to get rid of the repressor. There is, and hopefully some of these words will now become familiar to you, an inducer. And the inducer is complementary to an allosteric site. Okay, an allosteric site on each monomer of our tetrameric Lac repressor. So in light blue here, this is an inducer. The inducer will bind to the Lac repressor at each of the 4 monomers and will cause a conformational change. In each of the 4 subunits, 1, 2, 3, and 4. And what that means is the Lac repressor can no longer bind to the operator. And so the Lac repressor falls off. Of course, what that means is if we sort of translate that into our diagram up here, the Lac repressor falls off. So let's just draw it over here. Lac repressor falls off because it is bound by our inducer here, whatever that is. And then our RNA polymerase over here, which initially couldn't bind, now has full access to the promoter because the bit of the promoter that was being bound by the repressor, because it overlaps with the operator sequence, is now available. So RNA polymerase can now bind to the promoter, can now start transcription here, and we can start to produce our mRNA, and we can then make our gene products, our beta-galactosidase, our permease, and the transacetylase, which we're not going to worry too much about. And that means we can now, in our bacterium, import our substrate, we can import lactose using our permease, and we can then convert it, break it down using our beta-galactosidase over here into glucose and galactose. So, I hope that makes sense. The only thing to point out here is what is the inducer, which is quite a good question that I always get asked. And the answer to this is really clever. Evolution has sort of allowed this to happen, right? It's really cool that the inducer is lactose, which hopefully makes oodles of sense to you. In other words, if our bacterium is swimming around here very happily, if we have lots of lactose in the environment, so I'll just draw that here. If we've got lots of lactose around, we will then turn on transcription in our genome here. It's not linear, it's a circular genome, but the point still stands. We turn on transcription of our 3 genes, our LAC-Z, our Y, and our A, and we make the permease that sits in the membrane here and transports lactose into the cell. And we make the beta-galactosidase, which can then convert this into product, hydrolyze that glycosidic bond. So, just to recap, when we have lactose in the environment, lactose will bind to the repressor, which is sitting already bound onto the operator sequence, causing steric hindrance for the RNA polymerase, meaning that the RNA polymerase cannot bind to the promoter. Transcription is off. Lactose, when it's in the environment, binds to the Lac repressor on each of the 4 monomeric subunits at an allosteric site. This binding causes conformational change within the Lac repressor, meaning it can no longer bind to the operator sequence and it dissociates, it falls off, as you can see here. That allows RNA polymerase to have full access to the promoter and bind to it, and then we kick off transcription, which is what you did in lower 6th. RNA polymerase, phosphodiester bond formation, nucleophilic attack, joining activated nucleotides together, or deoxyribonucleotides together. Condensation reaction. So, hopefully that makes sense as to why lactose is itself the inducer, because that's the whole sort of signal molecule to say turn on the genes we need for metabolizing lactose. So, hopefully we've now covered induction. I'm just going to edit my diagram. Oh no, I've done that. Yeah, that's fine. Anything else I need to say on that? Don't think so. So, induction of the Lac Operon, tick. There is a really nice question here that I often do with my 6th form, and I tend to talk about it quite a lot because it's quite important. There is a glaring problem with this system. It's great. Lactose is good as a signaling molecule because its very existence is then telling the cell, in inverted commas, to generate the enzymes and the proteins required for its metabolism. But the problem is the very fact that lactose itself is the inducer. So, the problem here is if you have a cell, if you've got a bacterial cell swimming around, and we've got lots of lactose in the environment. How is lactose going to get into the cell? Well, we've talked about this. We need the permease, which is encoded by the LAC-Y gene. So in order to make the permease, which I'm going to put in the membrane here, to get the lactose in order to make this guy, we need to turn the Lac Operon on. We need to do transcription of our three genes, three structural genes, Lac Z, Lac Y, Lac A, to make, in this case, if red is Z, purple is Y, green is A, we need to make, in purple here, the Lac permease to be inserted into the membrane. But the problem is, in order to do transcription here, the repressor, which don't forget is this sort of tetramer, needs to be kicked off the DNA. And that is achieved because the lactose binds to it. So this very lactose here needs to bind to the repressor to cause it to fall off. Hopefully you can see this is a real chicken and egg situation. How do you get the lactose in to turn on the Lac Operon to make the permease that gets the lactose into the cell? How do you ever get started? If you need lactose in the cell to turn on transcription, you need to transport it into the cell. But you cannot do that unless you've turned on the operon, for which you need lactose to act as an inducer and cause a conformational change in the repressor. So this is a big, glaring sort of problem that certainly a lot of the students that sort of really think about this often come to me with. Their head is hurting and they sort of can't quite make sense of it. And the answer to this is quite simply that biology is not 100% efficient. This whole system is not 100% efficient. Which is not great, but what it does is it allows the whole system to get started. So what happens is, how do we get started? Well, even if the Lac repressor is bound, we get spontaneous dissociation of the Lac repressor. It will spontaneously fall off the operator. Fairly low frequency, but occasionally it does, just by chance. Because of course it is binding to the operator via non-covalent interactions, so ionic bonding, hydrogen bonds, London dispersion forces, that kind of thing. So all you need is for a Lac repressor to temporarily fall off the operator sequence, and what that means is you can then recruit RNA polymerase over here to do a little bit of transcription. It might not be for long, but it will be enough to put a few molecules of permease, maybe only one or two, let's say here, into the membrane. What that means is you can import now a little bit of lactose, maybe a few molecules of lactose here, not very much compared to what's in the environment, because we've not got much permease. But hopefully what you now appreciate is that the lactose here, we've got a little bit. That means it can go and bind allosteric sites on the repressor, conformational change, the repressor falls off the operator, and you do more transcription to make more permease. And what you start doing is then, depending on how much lactose you have, you make more and more permease to get more and more lactose into the cell, and bingo, you have now kicked off the process. Ooh, that is a weird smiley face. That's even, well, yeah, anyway. Hopefully that kind of makes sense. There is a sort of further question to this, but I'm not going to go into that. There's sort of another issue here, which is, you know, what happens essentially if there's no lactose in the environment, and you make a little bit of permease, well, surely no lactose will be sort of moved into the cell. What happens to the permease? But I'm not going to go into that. This is kind of the sort of most you need to worry about. You don't need to know about this really, but I did just want to mention it because those of you that really sort of think deeply about this topic will sort of hopefully have come across that conundrum before. How do you ever get started? So I'm just going to plug my charger in because I think my thing is about to die. Hopefully that has sorted. Right. So that is the induction of the Lac Operon done. I am now going to talk briefly, not very much, about the structure of the Lac repressor, just because I think it is useful to understand how this actually works. So this is point number 5. So how does this actually work? And this nicely links to your protein topic. Again, it's a synoptic link to lower sixth where you did protein structure. So if we zoom into the operator sequence, so just very quickly, very crude diagram here. I'm not going to do colors. You've got your promoter sequence here. The operator sequence overlaps with it. You then have your 3 structural genes in the case of the Lac Operon, and you've got your terminator sequence here. If we zoom in to the Lac operator sequence, what you will see is that there are 3 individual binding sites here for the Lac repressor. And we call them O1, O2, and O3. And they're in that order, just for information, but don't worry too much about it. So what happens is the operator has to bind. So the repressor must bind to 2 sites of either O2, O1, or O3. But it has to bind to O1. This is critical for function. So we have to bind to O1, and O2 and O3 are sort of synonymous. It doesn't matter which, we just have to bind to one of these. So we have to bind to O1 and either O2 or O3. And what happens is, if I just zoom back in on the operator sequence, you get this kind of a structure. So O2 is up here. We have O1 and O3. And the repressor, we said, is a tetramer. So 2 monomers bind, let's say this one and this one. 2 monomers bind to O1. So this is monomer number 1, monomer number 2. And the other monomers bind to O2 in a similar way. So we've got monomer 3 and monomer 4, and they are sort of connected together. Don't worry about how, but they're essentially connected. So it acts as a tetramer. But each monomer, 2 monomers bind to an operator sequence, and each monomer sort of interacts with the operator sequence. And what this does is it causes DNA looping, as you can see here. And FYI, it could also sort of bend around the other way and interact with O3, but don't worry too much about that. We'll just do O2 for now, for this example. And what happens is your lactose, let's do the lactose in blue, will bind to each of the monomers, cause a conformational change, and that will, if you have enough lactose to bind to all of the monomers, that will cause dissociation. Oops, what happened there? It will cause the repressor here to fall off. So if we're bound to lactose, here we go. The Lac repressor dissociates, changes its conformation, falls off the both operator sites. So of course, the 2 operator sites that we're talking about here, O2, O1. What that means is we now have full induction. So the operator is now fully sort of not tied up, binding or being bound by the repressor. So the promoter RNA polymerase can come along and can do transcription. So this will lead to transcription and turning on of the Lac Operon. But the key point here is that turning on the Lac Operon or turning it off is what we call bistable, which means it acts like a light switch. Oops, that's a horrible W. What we mean is we're either on or we're off. There is no middle ground. There's no sort of dimmer thing here where we're a little bit on. Okay, the Lac Operon is either fully turned off or fully turned on. This is achieved because of this mechanism, because of the DNA looping and because we have a tetramer. So looping and the fact that this is a tetramer, this repressor, both contribute to being bistable. And the reason is as follows. If I just come back to this diagram, hopefully what you can imagine is if we only have a little bit of lactose, so this was if we have enough, but if we don't have enough lactose in the environment, we have a small amount, small concentration of our inducer of lactose, you can imagine that, let's just do it this way for the sake of argument. Like that. You can imagine that our lactose binds here and here, and that causes dissociation from these two monomers, so monomer 1 and 2, from the operator sequence O2. But we don't, because we've got a low concentration of lactose, we don't have enough. There is not enough lactose to bind to these monomers. So these guys remain closely bound to the operator sequence O1. What that means is, as soon as your lactose levels are depleted, if you genuinely don't have much lactose in the environment, very quickly, these two lactose molecules will dissociate. They'll sort of leave. So from monomer 1 and monomer 2, off they go. They're sort of over here now. Try and do them in blue. And what that means is we can very quickly rebind to the operator sequence here. We're not having to rebind both operator sequences because we still remain attached to O1 because we didn't have enough lactose to dissociate those two monomers from O1. So what that means is you need a decent amount of lactose, or you sort of need enough lactose in the environment. And what this does is it protects from the fact that you could just have a little bit. If you're the bacterium, you don't want to waste resources if there's, one molecule of lactose. I mean, I'm exaggerating, but if there's only one molecule of lactose, you don't want to waste time, transporting it and metabolizing it because there's just no point. What you want is enough lactose in the environment to make it energetically favorable for you to actually turn on transcription, make the proteins via translation, and then import and then deal with the lactose as a substrate. So don't worry too much about all that, but I just wanted to point out that means the system is bistable. You need enough lactose in it to be imported. And I'm not going to give you the numbers on that. There's been experiments done that have determined how many permease molecules you need to get enough lactose in to actually fully induce the system and not just go back to being repressed very quickly. But that's fairly dull and not important for your exam. Cool. The final point, so that's how the repressor works. Well, actually, there's one more thing to say here, actually, which is how does the repressor bind to DNA? And this is what links to your protein structure from year 1. If I draw the Lac repressor as I've been doing like this, these are the two monomers here. We've got another two here, and they are sort of bound together. If you were to look at each monomer, so let's just zoom into this one here, what does it look like? Well, it's obviously a sort of mush of a protein. So in tertiary structure, it will sort of look a bit like this. But within this, there are important elements. And I'll just sort of draw it out here like this, just to make it clear. we enter into an alpha helix here, going this way, and it then loops and forms an alpha helix going this way. So when we draw a cylinder here, what I mean, of course, is that the protein is folding in an alpha helix here and here. This bit of floppy protein sort of amino acid sequence is called a turn or a beta turn. It's another type of secondary structure, but don't worry too much about it. These guys are alpha helices. And these are both examples of secondary structure. And of course, this may just sort of continue and fold back into the protein. But within the protein, you have this motif, this helix, alpha helix here with a turn and a helix. And so this whole structure in purple here, this helix with a turn and another helix, is called a helix turn helix. And it's called a motif. Don't worry too much about that. But the key thing is that you find this in many proteins, but it is somewhat superior to secondary structure. It is not just one alpha helix, it is an alpha helix linked to another alpha helix via sort of a turn, via this sort of bend structure of primary sequence. So this whole thing, this here is secondary structure, as is this. But the whole thing comes together to form a helix turn helix motif that is functional, tends to have a good function. So this whole thing we now refer to as a different step in our protein level of folding, and it's called supersecondary structure. Don't worry too much about it, but it's just useful to know. There are lots of them, lots of examples. A helix turn helix is one such example. And what function does it do, I hear you ask? Well, it binds to DNA. And it can do that because the bond angle here, poorly drawn, because it doesn't look like it, better drawn here actually, but anyway, point is, the sort of angle here between the two alpha helices is 120 degrees. Bit of pub quiz trivia for you there. And it can bind to DNA because of this 120 degree gap, and of course the alpha helices, because DNA looks like this. And you've got your hydrogen bonds between the base pairs, yada, yada, yada. And what you have here is you often have a small gap here. You can see that sort of small sort of recess here. So that's called the minor groove. And you have a much bigger one here. That's called the major groove. And they are just physical properties because of the DNA being a double helix. And the important thing here with regards to the repressor monomers is that the helix turn helix motif, the 120 degrees between the monomers, is perfect to straddle the DNA, and that's really poorly drawn. It's sort of one is one side, one is the other. So it kind of sits on the DNA in the major groove like a human sits on a horse. It sort of straddles the DNA. So a helix turn helix motif can bind DNA in the major groove. And it can do that because 100, as if by magic, 120 degrees here is the perfect angle with which to dock into the major groove of DNA. So if I now go back here, if we imagine our DNA is looping around, so I'm not going to draw the double helix just for ease, but the DNA, of course, is looping around like this because we're looping and it keeps going on in this direction. And we have our operator site in green here. So that's our operator site here. We've got an operator site here and there's one here, remember? So this was, from memory, this was O2, this was O1, this was O3. And we're interacting with the DNA because, I'm just going to get rid of that here, we have a helix turn helix. So we've got one helix going this way, that then links to the other helix going this way, poorly drawn with highlighter, that was a bad choice, but I'm going with it anyway. So helix turn helix there. This monomer also has a helix turn helix motif, trying to make the point about the 120 degrees, but never mind. And they're both sitting, so of course 120 degrees here, 120 degrees here, and they're both straddling DNA. This one's facing this way, this one's facing this way. Okay, just like I've shown here. It's literally sitting like this green one, into the major groove of DNA. And it's doing that on each monomer. And of course, the same thing's happening down here. So just to make it abundantly clear and take even longer to finish the video, but hopefully it makes sense. You can speed it up if you need to. Here are your alpha helices. So facing this way and this way, this way and this way. So that is how these monomers are binding to DNA. And I make this point because if you want the repressor to fall off DNA, so let's imagine this red squiggle is the repressor again. If we add the inducer, let's say the inducer binds to a site here. So this is lactose in blue. This is our inducer. How does it cause a conformational change and the repressor to fall off DNA? It does it because it causes a lot of changes within the tertiary structure, and it causes this this angle of 120 degrees to change. It actually causes these two helices to move further apart from each other and widen that angle. What it means is essentially the 120 degree angle that's required for docking into the major groove by the helix turn helix motif, if that's no longer 120 degrees, we're changing the angle between these two helices, we can no longer bind to the major groove, and this whole thing falls off. Whoops, not the DNA, obviously. Oh dear, what's happened? There we go. Oh well, the whole thing falls off anyway. Hopefully you get the point there. So that is how the Lac repressor works in terms of structure. Again, don't worry too much about all that, but I did want to bring in the idea, certainly for those of you looking at Oxbridge, of the idea of a super secondary structure, of which one of them, the helix turn helix, is found in a lot of DNA binding proteins. And it can bind to DNA because of the 120 degree angle between the helices, which allows it to dock into the major groove of DNA. And changing that angle means that that monomer that the helix turn helix is associated with, let's say these guys over here, these guys can therefore no longer bind to DNA. They can no longer bind to the operator sequence. And if lactose, the inducer, is bound to them, these helices, the angles change, and therefore we fall off. We dissociate from the operator sequence. So, quite a lot there. We are very nearly done. This shouldn't take too long. The final point, number 6, is to look at catabolite repression. And before I do that, I'm going to make one very important point. Actually, no, yeah, I'll do it now. The Lac repressor binds to the operator for the Lac Operon, stops transcription, and when we add an inducer, the repressor is inhibited, falls off, and we get transcription. So, hopefully, linking back to control circuits, what type of control circuit is the Lac Operon? Well, essentially, if you imagine our substrate here being converted into product, in other words, turning on the transcription is kind of what we're saying. This whole induction here, turning on transcription of our 3 genes, is inhibited initially by the repressor. And we turn on transcription because we have an inducer, lactose, which inhibits the repressor by binding to it allosterically. That means we are no longer repressing the system and it is turned on. And hopefully you remember from the notes on control circuits, because the repressor is the main key player, it is a negatively controlled circuit. And because when we add the component, we add an inducer, we turn on production of the product, it is negatively controlled inducible. Oops. So that links back to our circuits, and you do need to know that the Lac Operon is a negatively controlled inducible circuit. Right, 10 minutes hopefully or so, and this will be finished. The last point is catabolite repression. Which is there is a system superimposed. It's a superimposed system on top. Oop, my writing is going horrible. Of the Lac Operon. Here we are superimposed on top of it. And the whole purpose of this is to do the following. Is to preferentially use glucose as an energy source. Over lactose. That is the idea. We need to preferentially use glucose. So if bacteria have glucose and lactose in the environment, we want to use the glucose up first and then use lactose. And this leads to something called diauxic growth. Which roughly, looks like this. So when bacteria grow, they increase in number, they then plateau and then start to increase again. And it is this plateau that represents the shift from metabolizing, in this section here, we're using glucose as the main energy source, to then turning on the Lac Operon. So here is where the Lac Operon would be turned on, to then use lactose as the respiratory substrate. This is achieved through catabolite repression. And I'm not going to give you much detail here because it's really not that important. But I do have to finally draw this for one last time. So bear with me. We have the promoter. We have the operator, which overlaps with the promoter, don't forget. And I'm not going to draw the ribosome binding sites, but we essentially have our three genes. Like this. This is in DNA, so we have a terminator here as well. That's the terminator. And just to be clear, this is Lac Z. This is Lac Y. This is Lac A. We also have the Lac I gene here, so that codes for the repressor. And above all of this, so upstream of all of this, we have one more site, just to add further complication, which I'll do in blue here. We have one more site here. And this site is really important. It is the binding site for an activator. So remember, we have activators as well as inducers and repressors. And it binds to an activator called CAP. So I'm going to draw a CAP like this. And CAP stands for catabolite activator protein. It is sometimes referred to as CRP, but don't worry about that. CAP is the term that we tend to use for this. And essentially what happens is CAP binds to this activator site, and what it will do is it will physically interact with RNA polymerase, and it will recruit RNA polymerase at high efficiency, so very efficient recruitment of RNA polymerase. It will recruit RNA polymerase to the promoter, and then off we go. Assuming, of course, that the Lac repressor is dissociated because we have lactose in the environment. So the key thing is, how does this fit into glucose? And the answer is as follows. CAP will only interact, so let's put this into notes. CAP, the activator protein, only binds to its binding site when itself is bound by a molecule called cyclic AMP. So this stands for cyclic adenosine monophosphate. It's a nucleotide monophosphate. So just to be clear, CAP does not actually function by itself. So we therefore call it an apo activator. And hopefully that term is not new to you if you look back at the genetic circuits in lesson 1. When we bind to cyclic AMP, in yellow here, we then form a functional activator that can then bind to the binding site. So cyclic AMP here is a co-activator. You need binding of the apo activator to the co-activator to then form a functional sort of complex here that can bind to the activator binding site. Once it does that, it can efficiently recruit RNA polymerase instead of sort of relying on RNA polymerase sort of diffusing randomly and finding the promoter. CAP, the CAP cyclic AMP complex makes it very efficient and increases the affinity of RNA polymerase for the promoter. You don't need to know how. It is quite cool, but you don't need to know how. Speak to me if you're interested. Catch me at the end of a lesson at some point. And the magic of all of this is regulating cyclic AMP. You can regulate the level of cyclic AMP to regulate CAP's activity under different conditions. And this is where glucose comes in. If we have a high concentration of glucose, it has an inversely proportional relationship to cyclic AMP. What this means is we have a very low concentration of cyclic AMP. And vice versa is true. If we have low glucose concentration in the cell, we will have quite a high concentration of cyclic AMP. And this is the magic formula. This is the important stuff here. Okay, the inversely proportional relationship between glucose and cyclic AMP. The reason that this is the case is because glucose is metabolized via respiration. And the products that are released, different products through these many processes, glycolysis, of course, being the first step. Some of these products, let's say this guy, can interact with an enzyme. And that enzyme converts ATP, adenosine triphosphate, into cyclic AMP, and we release pyrophosphate. Don't forget that's 2 phosphate groups. So pyrophosphate refers to 2 inorganic phosphate groups. And I'll show you that in a bit more detail in a minute. This enzyme that does this is called adenyl. Oops, I can never spell it. Yeah, I think so. Adenylate cyclase. That'll do. I think that's how you spell it. Adenylate cyclase. And that's what this enzyme here is in red or green now. and it converts our substrate into our product, ATP, into cyclic AMP. Now, how does this happen? Well, if you have glucose around, we produce these products, and these products actually inhibit allosterically. So let's say this guy's in blue, kind of like lactose in a way, binds to this enzyme, stops it working, which gives rise to this relationship here, which is that if we have glucose around, we have lots of these products, which means that your adenylate cyclase enzymes are being inhibited allosterically, causing a conformational change in their active site, meaning that the active site is no longer complementary to the substrate, which means that we don't form much cyclic AMP. We only form cyclic AMP when glucose is depleted. Or certainly we form less of it when we have less glucose around. So before I tell you the structure of cyclic AMP, let's just get the main point out of the way. Hopefully this allows you to appreciate what happens. If we have glucose and lactose available to us, okay, we would rather, as you can see here, use glucose first as a cell. It's more energy efficient. We get more energy from it via respiration. So what happens is that because we have glucose around, not much cyclic AMP is going to be produced. So I'm going to delete this from here. No more cyclic AMP. That means our activator, the CAP protein, is an apoactivator, cannot bind to the activator sequence, and it therefore cannot recruit RNA polymerase. Don't forget, this is linked to RNA pol. It cannot recruit RNA polymerase to the promoter very efficiently at all, which means RNA polymerase, if it does bind to the promoter, will be completely by accident. It's not recruited. Of course, we've got lactose around as well. I said if we've got glucose and lactose around, lactose will be binding to the Lac repressor. The Lac repressor dissociates from the operator, and we turn on transcription of these three genes. But the problem is, well, the reality is, that's what I told you before. We have to now look at it in terms of CAP. In theory, lactose is around and binding here, and we have no repressor bound to the operator. We're turning on transcription. Except RNA polymerase is not very efficiently recruited to the promoter because we don't have CAP interacting with cyclic AMP and binding to the activator. So actually this doesn't happen very much. You might get a little bit of transcription, just a tiny amount, but not much. So essentially this doesn't happen and this system is off. But what's really nice about this is as soon as you use up all the glucose, this time lag here to turn on induction of the Lac Operon is very short. And the reason it's short is because you have lactose around anyway in the environment, the repressor here, oh, that's a horrible color. The repressor here is already bound to lactose because you had lactose around. So you don't have to unbind your repressor. It's already not bound, right? All you need to do is recruit RNA polymerase. And we can do that when the glucose runs out, because when the glucose runs out, we produce cyclic AMP in yellow here. And the cyclic AMP activates the CAP complex as an activator. CAP can bind to the activator, recruit RNA polymerase efficiently to the promoter, and we get lots of transcription now, and we get full induction of the Lac Operon. So you can imagine, though, if we don't have any glucose around, we only have lactose, we get full induction because this is happening anyway, right? If we don't have glucose, we've got cyclic AMP. That is allowing CAP to bind to the activator. We're recruiting lots of polymerase, and the repressor is bound. So we have loads of transcription. The other way to do this is if we don't have much CAP around. So we've got lots of glucose and not much lactose, which it means that if you don't have much lactose, I'm just going to sort of try and I'm trying to wonder how my diagram could get any more messy. So imagine if you don't have lactose. So lactose, imagine we don't have that, but we do have glucose, right? hopefully you can appreciate that if there's no lactose, there's no inducer. So the repressor is going to be bound fully to the operator, stopping transcription. And even if we wanted to recruit RNA polymerase, we can't, because if we have lots of glucose, we have less concentration of cyclic AMP around. That means this guy's gone, which means that the cap can no longer bind to this, which means even if we have polymerase floating around, we cannot recruit it to the promoter very efficiently at all anyway. And even if we did, it can't bind because of the Lac repressor binding to the operator. Right, that's the main point of the news. So I'm going to tick off now our final point. We got there eventually. Quite a tough lesson, that one, but there's a lot of information there. Probably takes a week or two to teach that in reality. So we've done that in just over an hour, I think, about an hour and 15 or so, I hope. I haven't checked the time yet. The final thing just to point out is, what does cyclic AMP look like? So it's all well and good just talking about this, but what is it? So let's look at ATP first of all. Hopefully you should be able to draw the structure. And I'm just going to go for a generic ribose sugar here, and the base, of course, is adenine. And let's chuck on our negatively charged oxygens, because of course it's an acid, so protons have been dissociated. And we've got our 3 phosphate groups here, inorganic phosphate groups. So just to be clear, for those of you I haven't taught before, this phosphate group is referred to as Pi, not P, because P means phosphorus as in the element. So it is not P, it is Pi, but don't worry too much. This is, of course, a ribose sugar, so we're not deoxy on the 2' or the 3' carbon, so we're OH here. This is the structure of ATP. We form cyclic AMP essentially because the enzyme adenylate cyclase will cleave this bond here, liberating these two together, and this is what we referred to as pyrophosphate earlier, PPI, pyrophosphate, 2 of them stuck together. And essentially, so if I now sort of just get rid of these, actually I probably could move it, couldn't I? Oh dear. Just to make it even clearer. So if I move that down here, there we go. So we produce the pyrophosphate, and essentially that does have an A- on it, so let's get rid of this. This phosphate then links up with the oxygen on the 3' carbon here. So the hydrogens are displaced. We then have the oxygen linking here, and I'll just draw that here. So that links to our phosphate group here, like that. Badly drawn, but you get the point. So this is the cyclic bit. And it's the link is between the first phosphate group, the alpha phosphate group, and the 3' carbon and its oxygen, forming cyclic AMP. Cool. Right, that's it. Long lesson for that one, but hopefully it makes sense. It is tough. It's one of the tougher topics at A level, but hopefully I've got into enough detail here to give you a really good overall understanding of what's going on here and why things are true. So yeah, any questions, do email me. I will see you outside of lessons, no problem.